chromosome. Now remember, most of you think that a circle is that. By the way, in Brazil that would be very nice, it means that. Never mind. That is not what I mean by a single circular chromosome. I mean that you take DNA, a long string of it that is wadded up inside the cell, and the two ends are joined. That kind of circle. So if you pull that circle out, it probably reaches to that door over there. Yes?
Sorry, what was the name of the gel? It was part of the...
Cytoplasm.
Can you say that one more time?
Cytoplasm. All right, so I'm going to go over the 5 parts again. Remember, it's recorded, you can listen. Cell wall, plasma membrane, the gel-like liquidy stuff that fills that everything floats in called cytoplasm, a ribosome, and a single circular chromosome. Those are the 5 parts of all bacteria. All prokaryote cells have the same 5 parts. Now there's some slight differences between the eubacteria and the archaeobacteria that we'll get to in a few minutes. But we're talking about the simplest cell now. And what used to be the only cell in the kingdom Monera, remember there is no kingdom Monera now. It was split into 2 domains and raised up above kingdom. It reproduces the same way all living things reproduce. Exact copy division. Exact copy division is mitosis. Now when your cells go through mitosis, they go through a whole bunch of phases. Prophase, metaphase, anaphase, telophase. And when it's between divisions, it's called interphase. Bacteria don't do that. They only have 2 phases, and they don't even name them. They just call it binary, meaning 2-step splitting. Binary fission. Fission means splitting. So they do go through exact copy division. What do I mean by exact copy division? If you start off with 1 cell with 1 chromosome, you'll end up with 2 cells, each with 1 identical chromosome. Start off with the same thing, you end up with. Exact copy division, you might more understand it as cloning. You're making an exact copy of what you started with, but now you have 2. So eubacteria do go through mitosis just like your cells to make copy. When you scratch yourself and you need more skin cells, they do mitosis through all those phases, and you get a new cell, exactly like the one that was there before. Bacteria do that. They just don't go through all those steps. They just basically go through telophase and interphase. So we call it binary fission. Some people say, since they're splitting like this, from one bacteria to that, they call it transverse fission. It means cutting it in half, transverse. All right, if I was to send you a multiple choice question on the midterm for saying what is the most important characteristic of a prokaryotic cell, you would say it doesn't have a nucleus. Wherever the chromosome is floating is called the fake nucleus or nucleoid. OID means fake. So if you're an android, you're a fake human. All right. So remember that nucleoid is just wherever the chromosome happens to be in the cell. So the nucleoid could be here, or here, or here, or here, or here, wherever the chromosome is. What makes it different from eukaryotic cells? Your cells have a double membrane around the DNA. And that double membrane surrounded region containing DNA is specialized and called a nucleus. Bacteria don't have a nucleus. Just push it. I know it squeaks. There you go. All right, so any question about the simplest cell on Earth? The prokaryotic eubacteria. Prokaryotic meaning microscopic, small, 5-part, smallest form of life on Earth. Eubacteria meaning the simplest of those 2 kinds of bacteria, the one with the 70S ribosome. Yeah?
Sorry, so just the prokaryotic, that's the one that has no nucleus?
All bacteria do not have a nucleus.
Okay.
No bacteria has a nucleus. And since all bacteria are prokaryotic, prokaryotes don't have a nucleus. All right, so let's move on to the second kind of bacteria. Remember that we didn't even know this kind of bacteria existed until a man by the name of Carl Stetter, who thought he would find the relatives of the first form of life by looking in environments that look like the early Earth. Remember, these are called archaic environments. The Earth does not look today like it did when life first evolved here. Remember, the universe is 13.81 billion years old. How do we know? Light travels at 186,000 miles per second. The light from the Big Bang we measured getting here, and we can go backwards and figure out how long it took to get where it is today. And the European Space Agency did a better measurement than we did. Our measurement said it was 13.5 billion years ago. They sent up newer equipment. Our equipment was in the 70s. They sent up a newer one. And just last week, the measurement came out, 13.81 billion years ago. All matter was concentrated into an infinitesimally, incomprehensibly dense dot that spun around and blew out in a spiral pattern. And at the moment of blowing up is when atoms came into being and light first appeared. And if you're religious, that was, in the beginning, there was light. Okay? All right. So, that's 13.8 billion years ago. Matter flung out. The first atoms came into being. The first light appeared. And when that matter was flung out, it continued to condense into little spiral things. And so a man by the name of Smoot, S-M-O-O-T, a United States guy, okay, from the USA, said, I think I can tell you about the Big Bang. I have an idea, a hypothesis, that if the Big Bang occurred, then if we take a picture of it, a fraction of a second after the Big Bang, the matter that's flung out will be flung out in that spiral pattern, but it will have hot spots because the matter is not flung out as a milkshake. It's matter being flung out in a pattern. And he said, if I take a picture of the universe a few seconds after the Big Bang, where those hot spots are where the galaxies exist today. So we sent up a satellite that went and stared back into the deepest recesses of space, back to the few seconds. Remember, light is a time machine. Light arriving now left there long time ago. So he found, it's called the relic radiation theory, and it proved that the Big Bang did happen and that he was right. It was flung out in a spiral pattern, and that where those little hot spots were a few seconds afterward are where the galaxies exist today. So after that, the next thing is one of those clouds condensed and exploded and flung out matter and created our solar system, and the Earth itself is about 4.5 billion years old. And from that movie that you saw, if you want to know anything about what I just talked about, you should have seen the How Life Began movie. I mean, not How Life Began, but In the Beginning movie on the origins, the one before the one I assigned. But anyway, when we investigate how long life has been here, they found a type of carbon that is only made by living things in rocks in Greenland that are carbon dated 3.8 billion years ago. So, 3.8 billion years ago, bacteria were here. And the conditions of early Earth did not look at all like they do now. The surface had water, but it was boiling ocean. The atmosphere was not oxygen, and all of what we have then, it was mostly methane, hydrogen sulfide, carbon dioxide, and carbon monoxide. And the temperature was very high, and there was carbon dioxide makes a thick atmosphere, so solar rays don't penetrate, so it was dark here. It was not bright, sunshiny blue skies, it was actually orange. And so, Carl Stetter said, if I can find conditions on today's Earth that are similar to when microbes first evolved, I bet we'll get a hint on the first forms of life. And that's why he went to these boiling water areas of Italy, this island, and collected bacteria that grew in boiling mud. And he sent them to Woese. And Woese analyzed their ribosomal RNA and found sequences never seen before. And so he said that bacteria that come from these archaic environments should be called archaeobacteria because they are bacteria. And they come from archaic environments. He further analyzed them and found that even though they have the same 5 parts, the parts are constructed somewhat different. The cell wall is not peptidoglycan, but pseudomurin. The plasma membrane still is formed just like all other membranes according to the fluid mosaic model. But in the phospholipid bilayer, the lipid is not cholesterol. In the eubacteria and in eukaryota, in the plasma membrane, the lipid is cholesterol. Which, by the way, is why you can't eliminate all cholesterol from your diet. Your cells couldn't divide. All right. What else did he discover? He discovered that they can't eat complex foods like disaccharides and polysaccharides. They can only eat monomers, and some of them have a heavy ribosome. So that puts them intermediate in evolution. They are still very small and have only a few parts. But one of their parts is like more complicated and related to eukaryotes. Also, he found these kind of archaea live in very bizarre environmental niches. And the way these bacteria all live in these very archaic environments, such as they live in acid. They live in boiling water. They live bacteria that live in water so salty nothing else could survive in. And there are bacteria that eat methane, hydrocarbons, and gasoline. All of these are very strange environments and very strange food sources and environmental conditions. And we thought they were just unique and interesting and amusing until people found there's a lot of potentially money-making ideas in the archaeobacteria. Because they live in such bizarre places, we can find enzymes in them that do things that we want done. And so these enzymes have become extremely economically important. If you Google extremozymes or products from the archaea, you will find there have been 300 products that make gazillions of dollars for the companies that have discovered them, all based on extremozymes, the enzymes of these bacteria that live in unusual habitats. And we talked about a couple of them. The biodegradable bleaches that we use today came from an archaea that had fallen into a tree with black bark, had fallen into a boiling spring at Yellowstone National Park, and the bark had been bleached crystal white, like the crystal you put up by your nose. That's called white. Okay. All right, and so a person noticed this and isolated this enzyme from the bacteria, and it became our biodegradable detergents that we use today, such as Spray and Watch, Clorox 2, and so forth. Another extremely important, probably, I'd say it's impacted probably 25, maybe 35% of our lives, is the fact that we were not able to sequence DNA until the archaeobacteria were discovered, because DNA is too small to read. So to read it, you have to make copies of it. But to make copies, you need a copy enzyme. And when you make lots of copies of something, you generate heat, which destroys DNA. So we had a conundrum. How do you make a lot of copies of DNA so you can read it without generating heat that destroys it? And someone said, why don't we use these bacteria that live in boiling water. I bet their enzymes do not destroy DNA. They are capable at working at these high temperatures. And so we got DNA sequencing, PCR, and all that from the archaeobacteria. So if you're asked, and you will be, what's the most important thing about the archaea that we discovered, besides the fact that they live in these unusual niches? It's their extremozymes that have been used from cleaning up oil spills to bleaching paper to reading DNA. Remember, whenever they have an oil spill, they try to get up the most of it, but they can't get it all up. And when they want to get it all up, they spray it with a bacteria that eats gasoline. Yeah?
What is extreme, what did you say? I can't see.
Enzymes from these bacteria that live in extreme conditions. Okay, they still have the same 5 parts, but some of them have a complicated ribosome. Their 5 parts are constructed slightly different. Remember, they don't have the same peptidoglycan cell wall, they don't have the same lipid in their membrane. And finally, they do copy themselves, like all other bacteria, by exact copy division, transverse or binary fission. So they are very much bacteria. They're still microscopic. They're just unusual and intermediate in evolution. And then the final cell, the one we're going to talk about 20 minutes today and 2 hours after the midterm when we talk about the eukaryotic protists and worms that cause disease, is the complicated cell. Remember, we're going to spend 2 hours and 20 minutes on the complicated cell in this class. And the rest of the 16 weeks, we're going to be talking about bacteria and a little bit about viruses. So remember, this is the cell that is all visible light. That's why it's called a true cell, because it's visible. It is the cell that makes up the 4 kingdoms that still exist. The 4 kingdoms being plant, animal, protist, and fungi. Cell has up to 20 different parts inside. And many of these parts have double membranes surrounded particular functioning regions. And when you have a region of a cell that is separated from the rest of the cell by a double membrane, and it performs a unique function similar to an organ in a human being, they call it a cell organ or organelle. So remember, there are up to 20 different organelles in eukaryotic cells, the most famous being, of course, the nucleus, followed by the mitochondria, and if it's a plant, the chloroplast, that long hollow tube that wiggles all around in here and connects to the nuclear membrane called the endoplasmic reticulum that makes lipids and carbohydrates. And some of the endoplasmic reticulum has bound ribosomes on it, so it also makes complicated proteins. A specialized region of the endoplasmic reticulum that makes storage bubbles is called the Golgi apparatus. The storage bubbles themselves are called vacuoles. So there are lots and lots of specialized regions or organelles in eukaryotic cells. They have a nucleus. That's the most important thing about them. They are complicated. They are the basic unit of eukaryota or eukarya domain. And they reproduce by exact copy division mitosis, but they go through all those phases. Prophase, metaphase, anaphase, telophase, and between divisions, interphase. Whenever you're asked to draw a cell, you are drawing a eukaryotic cell in interphase. You know, if I say to some 5-year-old, draw me a cell, and they'll, that's a eukaryotic cell in interphase. Why? It's got a nucleolus, a nucleus, and you've got a membrane around it. So this is the eukaryotic cell. All right, so what's the rest of this chapter? The rest of this chapter is about bacteria. And what we're going to do is we're going to look at every piece that we've ever found about every bacteria, And remember, no bacteria has everything we're going to discuss. This is everything we have ever discovered about bacteria. So, the first thing you need to know is shape. And if you're given two choices, the correct name for the shapes are cocci and bacilli, or round and rod. There are only two shapes, cocci and rod. Now, you can't be almost pregnant, and we're having a little bit of trouble with people that are already. You see, you can either be pregnant or not pregnant. You can't be almost pregnant. And so we have people that don't understand in English that English has a root word and then they have prefixes. And so if I say coccobacillus, it means it's a bacillus that's roundish. It doesn't mean it's a cocci. Remember, if you have something in front of a word, it modifies what follows it. So coccobacillus is a bacillus, which is a rod. Okay. Did you have a question? Oh, okay. Okay, so now, what if you're allowed different kinds of cocci, different kinds of rods? Then there are spirilla, which are wavy rods that are rigid. Then there are vibrios that are shaped like commas. Then there are corkscrew ones that have a sort of a, looks like a rigid, what would you say, bar down the middle of it. Okay, so remember, if it is wavy, it's spirillum, but if it's corkscrew shaped, it's spirochete. And then there are squares, rosettes, and stars. All these are rods. Now, why don't we ever talk about squares, rosettes, and rods? The reason is, it doesn't make us sick, we can't eat it, and we haven't found a way to make money off of it, so we don't study it. It's just America. Alright? So there are people in universities that study the rare forms from the Red Sea where there are squares and stars and rosettes, but generally speaking, we don't cover them in classes because they don't make us sick. If we find one that makes us sick, we'll start studying it. Alright, so if you have one cocci and it divides one tongue in one plane, then you have two and they're kind of pressed together so they look more like kidney beans than two circles. That's called diplococci. Does everybody know what planes of division are? Length, width, and depth. Remember, right now we're seeing in 3 planes, but that video is in 2, length and width. It shows no depth. You have to get a 3D camera for that. Alright, so if a cocci divides once in one plane and you have 2 cocci stuck together, remember they tend to stick together and push together so that they're not 2 round circles like this, like a figure 8, but they look like kidney beans glued together. And that's called diplococci. If you have one cocci divide a whole bunch of times in just one plane, so it looks like a string of pearls, that's called streptococci. And here's something confusing and very annoying. The word bacillus means rod and a genus. If it's capitalized, it means the genus Bacilli, which is a rod. Streptococcus is a shape in a genus. So if it's capitalized, it's a genus, where all of the bacteria inside are streptococci in shape. So remember, it depends on if it's capitalized or not, whether we're talking about a genus or a shape. Alright, so streptococcus is a whole bunch of cocci or spheres on a chain. The next one is, what if you have a cocci that divides one time in one plane and another time in another? So you get 4. Let's call it tetrad. What if you have a cocci that divides one time in all 3 planes, length, width, and depth? Then you get a cube, and that's called sarcina, and it's the name of a genus as well as a shape. So if you remember, one of the answers on our practice quiz was sarcina, with a capital S. That's the genus. What shape is it? Cube. A cocci in cubes. All right, if a cocci divides many times in all three planes, so it looks like a whole bag of grapes, then that's Staphylococcus, and remember that's a shape and a genus. It's annoying. All right, and then of course we already talked about rosette stars and squares and all that. All right, so in chapter 4, what are we going to cover? These are the major topics. These are the pieces of bacteria from the outside to the inside. So if we were walking toward a bacteria we didn't know, and we were going from the outside to the inside, this is the organization that we're going to go through. From the outside to the inside, what are all the things we've ever discovered about bacteria? And the first thing we're going to talk about is things outside the cell wall, called external structures. And of external structures, there are 4. If it has a paddle and can move, is motile, it has a flagellum. A hollow protein tube used for sex is called a pili or conjugation pili. And by the way, unlike us, they can have multiple pili and have sex in several different places at the same time. So you can see 2 cells with like 5 pili connecting them. Nasty. I'll show you some bacterial porno a little bit later. All right? And then a hollow tube used for breathing through, like a periscope, or holding on, is called an attachment fimbriae. And then finally, if you have a complex polysaccharide all the way around the outside of a cell, and remember that's a sugar, it's called a glycocalyx. If it's highly organized and solid, it's a capsule. If it's not, it's a slime layer. But it's all called glycocalyx. All right, so these are the things outside the cell. If it can move, it has a flagella. And there are 4 ways that we can tell if something has a flagella. 1, we can do the flagella stain, which is we make a smear, and then you put a coating of something very similar to India ink on the smear and let it air dry and wash it off and coat it again. And you do it 30 times. It's called the flagella stain, and the technique for making a flagella visible is called the mascara technique. You see, none of these girls have these beautiful lashes they're wearing. In the morning when they wake up, they don't have those. They get it out of a bottle. It's called mascara, and it's actually ashes or soot plus a quick drying glue. And so you put it on, and it gets longer and thicker with each coating. And that's what we do to make flagella visible. So one of my midterm questions will be, the mascara technique is used to detect what characteristic of your unknown bacteria? Motility, to see the flagella. Okay, how else? Who can name me the other 3 ways that we detect motility?
Gel stab. The gel stab.
Gel stab, right. Remember that if it's motile, there are 3, drugs. There are 3 ways in which the gel stab can be done. You get the needle, you put a little bit of bacteria on it, you stab it directly down the middle, 3/4 of the way deep and back out, and put it in the incubator and come back in a day, and you will have a dark red line down it. You will have a dark red line with some peach fuzz, or you will have nothing. If you have a dark red line and the gel outside remains the same color, that is growth but non-motile. No flagella. If you can see the red line, but it has fuzzed away like they swam away, because when they swim through this, it reduces it and a pink color appears, then this is called growth with motility. It means they have a flagellum. Finally, if there's not a sign at all in there, it means your needle was too hot, or your bacteria is dead. Dead things don't move. Did you know that? Dead things don't move. And this is called the motility gel stab, or motility deep. And that's the best method of detecting motility of all our tests. Okay, what's another one? Hanging drop, right. You loop dip into a broth overnight, and the next day you bring it back and you mix it, and you put 10 loopfuls in this little slide that has an indention in it, and then you cover it with a cover slip that you put Vaseline on, and then you put a drop of oil and you try to focus it with as little light as possible. I'm looking to see if I can find one of those hanging drop slides, because every once in a while somebody can't get theirs right. Just FYI for fun, pass this around. That's a fluorescent antibody slide there. It has just sort of little regions to do the drop of fluorescent antibody. Aha! I know, I'm sick. Don't worry, I'm under treatment. There. All right, I'm going to pass a couple of these around. I want you to okay. Pass one back to that row behind you there. Feel the hanging drop slide. You're going to put 10 loopfuls in there. You're going to put 4 dollops of Vaseline on the corner of a cover slip. Put the cover slip on there, add a drop of oil, put it on your microscope, bring your condenser up, get your 10x into place, get everything ready, turn your light on very low, and you got 3 minutes to focus it. And if your bacteria is motile and has a flagella, it will be tumbling and moving around like that. If it's doing this, it's just Brownian motion, and it's non-mobile. After 3 minutes, it'll be dead. It's cooked. Hey. And actually, I'm looking to last in about 2 minutes. Okay. Oh, and I did get the what's your name? I asked for that order for you to go to Friday. No, just for Friday.
Oh, we already saw it. We already saw it.
Oh, anyway, I asked that you could access it, and then I called. I'm just giving a review for how to touch it. Just from 8 to 11. All right, so see you there. All right, so that's the third kind of motility. Let's see who's a smarty. Oh, no, I'm kind of scary about asking that. But anyway, what's the last method in that we talked about briefly of detecting motility in your unknown? Anybody remember? Nope, we talked about hanging drop, motility, stab.
Ultraviolet. There's a
What?
mascara.
What? Mascara, flagella, no.
What
There's
now?
one more.
What now?
Here it is. Plate observation. If you get an auger plate and you put your bacteria, a drop of it, it will swim away overnight and make steps. Chromosome. Now remember, most of you think that a circle is that. By the way, in Brazil that would be very nice, it means that. Never mind. That is not what I mean by a single circular chromosome. I mean that you take DNA, a long string of it that is wadded up inside the cell, and the 2 ends are joined. That kind of circle. So if you pull that circle out, it probably reaches to that door over there. Yes?
Sorry, what was the name of the gel? It was part of the...
Cytoplasm.
Can you say that one more time?
Cytoplasm. All right, so I'm going to go over the 5 parts again. Remember, it's recorded. You can listen. Cell wall, plasma membrane, the gel-like liquidy stuff that fills that everything floats in, called cytoplasm. OID means fake. So if you're an android, you're a fake human. All right. So remember that nucleoid is just wherever the chromosome happens to be in the cell. So the nucleoid could be here, or here, or here, or here, or here, wherever the chromosome is. What makes it different from eukaryotic cells? Your cells have a double membrane around the DNA. And that double membrane surrounded region containing DNA is specialized and called a nucleus. Bacteria don't have a nucleus. Just push it. I know it squeaks. There you go. All right. And when it's between divisions, it's called interphase. Bacteria don't do that. They only have two phases, and they don't even name them. They just call it binary, meaning two-step splitting. Binary fission. Fission means splitting. So they do go through exact copy division. What do I mean by exact copy division? If you start off with one cell with one chromosome, you'll end up with two cells, each with one identical chromosome. Start off with the same thing, you end up with. Exact copy division, you might more understand it as cloning. You're making an exact copy of what you started with, but now you have two. So eubacteria do go through mitosis just like your cells to make copy. When you scratch yourself and you need ribosome and a single circular chromosome. Those are the 5 parts of all bacteria. All prokaryote cells have the same 5 parts. Now there's some slight differences between the eubacteria and the archaeobacteria that we'll get to in a few minutes. But we're talking about the simplest cell now. And what used to be the only cell in the kingdom Monera, remember there is no kingdom Monera now. It was split into 2 domains and raised up above kingdom. It reproduces the same way all living things reproduce. Exact copy division. And exact copy division is mitosis. Now when your cells go through mitosis, they go through a whole bunch of phases. Prophase, metaphase, anaphase, telophase. More skin cells, they do mitosis through all those phases, and you get a new cell, exactly like the one that was there before. Bacteria do that, they just don't go through all those steps. They just basically go through telophase and interphase. So we call it binary fission. Some people say, since they're splitting like this, from one bacteria to that, they call it transverse fission. It means cutting it in half, transverse. All right, if I was to send you a multiple choice question on the midterm for saying what is the most important characteristic of a prokaryotic cell, you would say it doesn't have a nucleus. Wherever the chromosome is floating is called the fake nucleus or nucleoid.